So a lot of new applications and data that has been developed at different resolutions and that is open access, is available for anyone in the world that has the capacity to compute and work with all these data sets. And it's not only about hydrological characteristics. There is a lot of data now that is related with land use and biological data that we can integrate. And this is a comparison of two data sets. For example, this is the Hydro Atlas, one global data set, and one data set from the United States that maps rivers at local scales. And this is mapping the distribution of farm fertilizer across the entire country. And you can see that there are definitely similar patterns that we can look into this. And when we look at these patterns, we need to ask, okay, these patterns that we're seeing at these scales are actually predicting similar things in terms of how they could affect the aquatic systems that we're analyzing at more local scales. Another problem here, but the problem is that when we are working with these data sets, you would look that there are data errors and where there is uncertainty in these applications of these global scale data sets. So there is a lot of discussions now in terms of how we should look also into the uncertainty of the application of these data sets at global scales to map biodiversity, but also to identify the threats that are affecting that biodiversity. And we are doing a review at the moment looking at how many of these studies that are actually looking at restoration, for example, at the catchment scale and looking for prioritization or identifying which areas could be affecting this aquatic system. Are there any of those studies actually accounting for uncertainty in any form? And you can see that most of the studies are not actually considering the uncertainty of using these large-scale data sets to map threats on aquatic biodiversity. So we need to look at these scale mismatches in terms of the use of data. We need to explore when this data integration between the different data sets is good and could be extrapolated to other areas. And hopefully, when we look at these different, how this data can be integrated, we can identify if the decisions that we make with one type of data in one region could be applied to a different region, right? So in this study, basically we try to identify one of these problems by looking at one of the main threats of aquatic systems, that is the flow of nutrients that is affecting freshwater systems in different regions of the world. And we evaluated the suitability of large-scale data sets to estimate these nutrients. This is part of a project that is looking at the effects of different impacts on the impacts of different anthropic threats from the land on aquatic system, and this is looking for how we can prioritize for areas that are good for restoration. So basically we compared the effects of global data sets, how we can map different threats at these large scales, at the river level as well, at different and multiple scales compared to local data sets. And what you can see here is that the model estimation, actually the performance of the models that use the global data sets compared to the local data sets is actually very good for some regions. So this is an example in the United States using one global open access data set that has a relatively coarse resolution, the Hydro Atlas, compared to a data set that is a local and that is used by the US government to map nutrient loads across the entire country. And we did it as well for New Zealand and for other regions where I'm just showing two examples today. And when we look at New Zealand, the results are not as good compared to what we saw in the US, right? And in this case, the performance of the model actually predicting those areas where we have more effects of nutrient loads on aquatic systems, it actually declines a lot. So why are we finding this? And it's actually not related to the type of data that we are using, because in both regions we are using the same spatial data to try to predict the same variables. The problem is actually with the calibrating data. the water monitoring data that we are using to calibrate these models is not about the spatial resolution of the data sets, but we are looking at how well the monitoring stations are actually representing the domain of the different environmental variables that we are using to develop these catchment models. So very good in the US, not so good in some areas of Brazil and New Zealand, for example. So what we are finding is that the spatial resolution of the predictors is not as important as the actual data that we use to calibrate these models. So what we are doing now is generate this open access harmonized data set where we are trying to get all water quality monitoring stations for different regions of the world. This is part of a work that one of my students is doing. And we are collected information from more than 24,000 stations, more than 1 million records of water quality monitoring data for the entire world. So please get in touch if you would like to collaborate, because we have some data gaps in some areas, especially in Africa and Southeast Asia. So in general, these models are good to understand the relative influences of threats on aquatic systems, generate hypotheses and identify gaps, and prioritize at large scales. And this is something that we really need when we are thinking about the frameworks of the global restoration for the 2030, for example, where we are actually prioritizing for hotspots of biodiversity or at catchment scales. But they are very subject to the spatial distribution of these monitoring stations and the temporal frequency of those observations. So there are a lot of opportunities to actually linking these local scale monitoring data with hydrological variables and socioeconomic variables to understand how much do we have to invest to restore aquatic biodiversity by identifying those areas that need our attention. Definitely these models can help us with that. And we have a lot of data that could be used for that purpose. But this is the key obstacle here. It doesn't matter how good a spatial resolution we have in terms of our data if we don't have good monitoring data at local scales that allow us to calibrate these models. So a lot of potential, but we also need this mix of local and large-scale models. Obviously, I'm showing you here what could happen with the catchment models at regional scales. These models can be downscaled to identify what are the main threats in these areas that need to be managed and restored to protect aquatic systems. But obviously, all the things that we are doing here come from the modeling perspective, but as always, this is something that I always want to highlight in terms that how much you want to downscale your data will depend on the objectives that you are consulting with the key stakeholders and the local communities in these areas. So thank you very much. This is a joint effort with institutes from the US, Brazil, and Australia, and Canada. And yeah, thank you very much for listening, and happy to answer any questions.
Thank you, Felipe. What is your take then? Is it possible to do global predictions if you have, for example, the map you show with water quantity, if the locations are so clustered in some places, so and you have these big gaps? What's your take? Is it possible? I mean, is it possible? You put that into a model and you get a prediction, but is it worth doing, or you just go for just local predictions and leave the gaps empty?
So I think it's possible to do the predictions as long as you have the water quality monitoring data that could be used in those areas. And the good thing about all the hydrological datasets, including the one that your group has developed, is that you have combinations of environmental data across different regions that actually allow to make those predictions. So I think it's possible to make them. The problem is with the level of uncertainty that you will have depending on if you have water quality monitoring in those areas or not. So that's why we're having problems in countries like New Zealand or in areas like Brazil, where there is not enough information of water quality, is very sparse, and then it's very difficult to actually get good models that have like good certainty. But in terms of the environmental data that is available, I think that it's a field that is developing very quickly, and we can actually use those data sets, those global scale data sets. Yeah.
Thank you, Andres. I have a question. One of the major threats to freshwater biodiversity is the loss of connectivity. Have you considered this variable for modeling, or is it possible to include in this activity? Because for restoration, I think it's a big step to include.
Yep. So in these models that I showed you, it only considers, so these are spatial explicit models that look at how the different nutrients or sediments are moving from the land to the water. So there are variables that are specific for how much you would expect to move to the water system. And the cool thing about all these hydrological data sets is that they actually show you the flow pattern, right? Like you see how connected are different streams, and you can manage through functions of stream decay or reservoir decay, how those nutrients are actually moving across the entire system. So these are spatial explicit models that allow you to do that. There are other types of connectivity that we haven't looked into here, like connectivity from wetlands, for example. That's very important, right? like groundwater connectivity, that it's also key to consider in these restoration projects. But at least in this kind of data, you can do like surface connectivity in a relatively, it's not simple, but it's something that is possible to do with these data sets. Yeah. And I think it's possible to do it as well with the data sets that were shown like in the previous session as well.
Yeah, yeah, I have a question. Thank you for, good presentation. My question is focusing on how do you take, how do you consider the impact of those sedimentation, siltation that goes to water when you want to look at the biodiversity as fact, like you have the freshwater biodiversity, and what you have been doing is just from the landscape, but how do you measure the impact on biodiversity? Do you get kind of a decrease in the species richness, or do you, that is what I want to know from the...
Yep. So one part that I didn't show here is that we are linking this data with actual remote sensing and in situ monitoring of, for example, algae blooms or sediment loads or turbidity in these systems. So then you can link the amount of algae blooms or chlorophyll A in these areas with some relationships that are well known with biodiversity. So we are not actually measuring species richness per se, but we can rank the different systems in terms if they are oligotrophic or eutrophic systems and compare with reference conditions. This is still a large-scale global data set, and this is something that we are doing at large scales. So we need to have like those relative rankings in terms of, so it's mostly based on water quality. Yeah.
Thank you again. And now we have a virtual contribution from Eren Turak from Australia.
Good afternoon, everyone. My name is Eren Turak, and I am a principal scientist working for the Government of New South Wales in Australia. I'm also the co-chair of the Freshwater Biodiversity Observation Network. Today I'll be talking about the Freshwater Biodiversity Forecasting Tool. We developed this to support spatial planning, inclusive of freshwater biodiversity in New South Wales. The extent of the modelling exercise we undertook was the entire catchment of the rivers of New South Wales, covering an area of about 1.7 million square kilometres, and that's roughly 1.5 times the size of Colombia. The tool incorporates multiple datasets, macroinvertebrates, fish, various tools and models developed at different times for different purposes. And all of these are integrated into a platform in a modular structure where we have 6 different modules. We've so far developed 5 of these. The last one is scenario evaluation, which we are currently working on. So each module generates outputs that feed into the next module, or the final outputs, which are biodiversity conditioned surfaces, spatial layers, benefit surfaces for conservation actions and restoration. At the core of this is macroinvertebrate data we collected from over 3,300 river sites over a period of about three decades. And we use predictive models to assign a biological integrity score, biodiversity condition score at each sampling site. And then we use this to extrapolate condition across the entire area that the tools cover. So we have 2 tools at 2 spatial scales. One is the broad scale with large spatial units, a total of about 13,000 catchments, and using disturbance data and environmental data from the hydrographical database. The other is a fine scale tool with covering a smaller area. The area shown here on the right is the far north coast of New South Wales, 31,000 square kilometres, roughly the size of Belgium, and we use national data from the Wild Rivers project. So that allowed us to map condition across an area, but we needed to also incorporate irreplaceability of individual catchments, and we used the bottom-up river typology. And this typology was developed based on data collected from individual river sites. We have one for macroinvertebrates from river edges, from riffles, a fish typology, a diatom typology, and abiotic typology. Here, I will only be talking about the results from the fish typology, which we generated using data from 1,500 fish sampling sites across New South Wales. And the typology has 6 river types. Here you have a brief descriptor, a photo of an example river, and a photos of the species that characterize this type. And these types, the species composition overlap with each other, and each of them sharing each pair sharing at least one species. So we use this information to generate a similarity matrix, which allow us to identify or calculate the contribution of each river type to regional biodiversity. And this allows us to generate, calculate regional persistence of biodiversity, where river types that share few species with other river types contribute more to the regional biodiversity. Then what we do is to take each subcatchment and modify, hypothetically, the condition to generate a surface or a spatial layer that represents spatial patterns in the relative contribution of each subcatchment, in this case 13,000 or 12,000 for the fine scale model, to regional biodiversity. So we have two scenarios, one in which we degrade the river condition at each site and then calculate the regional biodiversity and rank all subcatchments in terms of their relative contribution to regional biodiversity. So the outputs are benefit surfaces. Here is for the broad scale model, a catchment restoration, benefit surface, and a catchment protection. So the spatial patterns you see here reflect essentially for the catchment restoration, areas of experience extensive forest tree clearing, areas of irrigation, coastal areas where high population comes out as being most important. Situation for the protection priority for the region is slightly different, where the disturbance layers don't influence the overall pattern so much. You have two zones, one roughly corresponding to the 600 millimeter rainfall, annual rainfall, and this really reflects the spatial area. The western part of this reflects the spatial area covered by one river type, the western lowland river type. In this case, the patterns, spatial patterns in the surrogate features dominate the overall pattern, where the eastern area essentially has higher protection area, but there is variation at a smaller spatial scale. When we compare the restoration priorities for the small area, the Northern Rivers region, what we see, this is a comparison between the outputs of the broad scale and fine scale model. What we see is that when we look, for example, at river valleys and protected areas, the fine scale shows greater nuance. And within protected areas, you have high and low restoration priority. And this is partly because of the averaging that comes from a coarser scale. For the broad scale model, the average subcatchment size is 134 square kilometres, whereas in the fine scale, only 2 square kilometres. But it also comes from the fact that we've used different disturbance data. For example, in this region in protected areas, you have a huge number of derelict mines, you have a high density of forestry roads remnant from before the time that it became a protected area. So these data sets provide greater insight and resolution into the condition and the action priorities. So where do we go from here? This is just an exploration of the tool in terms of how we can use it to inform decision making and reporting. What we've seen is that two different spatial scales can be combined in a single platform where we use essentially one coding language. In this case, we have two versions, one in R, one in Python. We use similar workflows, data pipelines to generate outputs. So the next step would be to create a continuum of spatial scales where you can pick and choose your most appropriate spatial scale for your different purposes. There is a huge collection of pressure and driver data sets available at various spatial scales. but most of these are not suitable. Essentially, there's a bit of work to do to sift through all the potential, the possibilities, combining global data sets with local data sets to generate the appropriate input data. For site condition metrics, we need to combine different types of measurements into one metric. We've so far only used macroinvertebrates, but that probably provides one view of local freshwater biodiversity. The models we use to extrapolate from individual river sites to the entire regions can be improved. At the moment, they're explaining quite a small proportion of the variability. We need to explore different modelling techniques. For the biodiversity features, we use the bottom-up typology, just one river typology. We need to explore others and probably also explore the use of top-down typologies and how we can combine with the bottom-up typologies. Finally, the integration we looked at focused on data and platforms. We need to also integrate concepts. In this work, I work with colleagues working in terrestrial biodiversity, so there's been a lot of combining of, especially use of some of the recent techniques developed for modelling and reporting terrestrial biodiversity. Thank you very much.
Okay, I think to stay in the time schedule, we continue with the next presentation, and that is from Spain, from Lide Izeta-Zalduendo. And yes.
Good afternoon, everyone. I am Lide Izeta-Zalduendo, and I come from the University of Navarra in Spain. I'm going to talk to you today about the impact of river fragmentation on freshwater fish species distributions, and we implemented this. We studied this through modeling. So this study we did inside a bigger project which is called Connect Fish with some colleagues from the Hydraulics Institute of the University of Cantabria. So for a bit of context first, as previous speakers already highlighted, the 1/4 of the world's freshwater fish species are threatened with extinction, and among these, almost half of these species are threatened by dams and water management. So man-made barriers like these dams affect riverine fauna by altering the river habitat and physically blocking the movement of species upstream and downstream the river. This is especially harmful and especially obvious for migratory fish species which need to move along the river network to complete their life cycles. So we wanted to study how this river fragmentation impacted the fish species, and for that we focused on the brown trout, which is a migratory species present in mainland Spain. And we studied this impact through the species distribution models, which, as you know, are tools that allow to explain and predict the distribution of species based on environmental predictors. So through where a species is and isn't, we are allowed to, and the relationships to environmental predictors, we are able to, for example, infer environmental relationships, which is the application that we studied. So yes, so the aim of this study was to model the distribution of the brown trout considering the barriers present in the river network in Spain. So for this study, our study area was mainland Spain, and we had a river network with more than 400,000 river segments, and each river segment had environmental data associated, like in previous talks other speakers talked about. So, for example, we had information about temperature, precipitation, and conductivity, and other kinds of environmental predictors. And so we used climatic, hydrological, geological, and topographic variables in our models. And as for presence-absence data, we extracted occurrence and sampling data from the Spanish fish chart, which is one of the most, I think, complete databases for freshwater fish occurrence data in Spain. And it was compiled by the efforts of the Iberian Ichthyological Society. And so with this data, we were able to extract 4,344 presence data associated to river segments, and the same amount of absences also associated to river segments. And then we also extracted barrier data from European, the database from the AMBER project, which has already been talked about, and national and regional databases, which were also incorporated later to the hydrological river network. So after all this data acquisition, the data was prepared for the modeling. The presence-absence data was partitioned into a training dataset and two test datasets to test the individual models and the ensemble models, which were the final product of the work. Well, the environmental data I already talked about, and then the barrier data was used to calculate some fragmentation variables. For example, information about, for each river segment, we had information about the number of barriers upstream and downstream said river segment. And one of the important variables that I want you to remember is we had one that was the barrier-free distance, which was the distance from the nearest upstream barrier to the nearest downstream barrier for each river segment. So with that information, we built two kinds of models: base models, which only included environmental predictors, and then a set of models which incorporated the fragmentation variables that we had calculated. And then we compared those two types of models through the performance of the models, for which we used two evaluation metrics, which the Trusky statistic and the AUC, the area under the rock curve, which evaluated the discriminative power of the model, which is the ability of the model to distinguish between a place where the brown trout was and where it isn't. Okay, so we got those metrics, and then we also checked the variable importance, which variables were driving the models that we selected, and a potential distribution map, and also the response curves for the variables that we used. Okay, so for some results, our base model was an excellent model in terms of AUC, more than 0.9. And the Trusky statistic was also good, although not excellent, more than 0.6. So you can see that the brown trout, according to our models, is mainly distributed in the north of Spain. And the variables that were most important in driving the distribution of the brown trout were, I don't know if you see it very well, but it was the precipitation, which is the most important variable, the mean temperature, and the mean daily flow. Okay, so now on to the comparison to the models which incorporated fragmentation variables. In this box plus, you can see the difference between the base model AUC and the model with barriers AUC, and the same for the Trusky statistic. So at first we were happy because we saw that the models with barriers, indeed, the performance of these models was better than the performance of only the base models. And these differences were statistically significant. However, and the model with the best performance was the one that incorporated the barrier-free distance, which is the one that I told you about. So, and actually this variable was the fourth most important variable selected by the model. So it was what we expected. And until then, everything was fine. But then we checked the response curve, which is how the predicted probability of occurrence varies with the variable. So we see that as the barrier-free distance increases, so the distance that the brown trout has available to move along the river network increases, the probability of presence of the brown trout decreases. So this was counterintuitive. It was the contrary of what we expected. And what we think is happening here is that, well, this is a map of mainland Spain and the river basin management units. And as we saw earlier, the brown trout is mainly distributed in the north of Spain, which is where rivers are shorter and they are more densely fragmented. So in the places where we have a greatest barrier-free distance is in the center and south of Spain, where the brown trout isn't present, but not because of barriers, because of other environmental predictors. So this is what we think is happening here, that our variables aren't capturing the fragmentation, or are rather explaining something else. And this is why they respond in a different way than we thought they would. Okay, so the key messages to take home are that the brown trout distribution is mainly conditioned by precipitation, temperature, and water flow. And adding fragmentation variables to brown trout distribution does improve the models. However, the variables that we use to assess the degree of fragmentation don't seem to be informing of what we thought of fragmentation, but rather underlying environmental variables that are confounding our results. So yeah, this is all. Thank you so much for listening. If you have any questions.
Thank you, Lide.
Oh, yeah,
Thank
sure.
you. This was really interesting. To follow up on your comment that you made about, well, that in the north you have the brown trout, in the south not, or in the rest of Spain not, and that this might confound your modeling results. Do you think you could split up your data set into river types or biogeographic regions to get rid of this, let's say, artificial structure in your data set?
Yes, in fact, what we were planning on doing next was do more of a local scale analysis to see if the environmental predictors like precipitation and temperature stay more equal, and the relationships to other less important environmental variables are more highlighted. So yes, I think so.
Thank you. Thank you. I was curious to know if the time of collection of the species was controlled for when the barriers were there. I mean, I would understand the analysis if the collection of the species was done after the dam was constructed, so you can find a relation. But if the samples were before, then the habitat has nothing to do with the barrier there, and maybe that's why the pattern doesn't make sense.
Okay, so the presence absence data that we used were from the year 2000 onwards, and there were few barriers built from the 2000. So yes, so.
Thank you again for the nice presentation. And we will continue now with Laban Musinguzi from Uganda. And yeah, the stage is yours.
Yeah, thanks. My name is Laban from Uganda. This is a national case study of how we're integrating species occurrences with different spatial data sets and conservation tools to make decision support tools. Okay, thanks. Okay. I thought that I should first connect with you with where I come from. It's my first time on this continent, and I'm from Uganda, which is a beautiful country known as the Pearl of Africa. And if you have not visited yet, I invite you to visit. And because I'm talking about fishes, I'd like to let you know that we have a lot of interesting fishes called cichlids, which are very beautiful, and they occupy most of the African Great Lakes. Now, a little background on why we are doing what I'm talking about today is because Uganda is a developing country. There is a lot of development that is going on. But unfortunately, it is a threat to aquatic biodiversity in the fresh waters. And what makes it worse is that even the national policies do not match the magnitude of the threats. But why is that so? It is because we have limited access to data to guide effective policies. Therefore, our work is addressing three problems: a problem in development, a problem in policy, and a problem to access to data. Now, our technical solutions to these problems are mainly two. That is aggregating all data that is available on the fishes of Uganda, and then using this data to generate decision support tools from it. If you were in the plenary there was a good presentation about data, and we think that this is an important work to address the challenges. So first, how is our efforts in data mobilization? So by 2017, we were on the left of this slide. All our data was in accessible formats. But since then, with support from different projects, we have been able to digitize all our data. It is now in accessible formats and in global platforms where it is available and driving research. So when we come to integrating this data with the other conservation tools and the spatial data, we begin by integrating our data on species occurrences with the information from FishBase. For those who do not know, FishBase is a global database with information about fishes. And when we go to FishBase, we get information on biology and distribution of different fishes. And when we integrate all that information with the spatial data for mapping distributions of fishes from the IUCN, we get refined distribution maps as our first decision support tool that can help decision makers to make decisions to guide development. Now, when we get the distribution maps, which are by species, and integrate them with the information on regional assessments, specifically those are national assessments, we generate a national red list, and that is also an important decision support tool. This bar graph that you're seeing is comparing conservation status on our national red list with conservation status on the global red list. We see that the national red list is better in informing decisions compared to the global red list. That is because it is based on more information that people who do global assessments do not have access to. We continue beyond the national red lists and try to integrate this information, now the distribution and the national red list, as well as the global one, to generate key biodiversity areas which are global, the best areas that we think are the best for conservation of specified species. And this is another tool that is important in guiding development, because these key biodiversity areas can be excluded from development or guide better mitigation measures. So from all this information so far, we have 7 sites which have qualified as key biodiversity areas and are now under review within the global KBA portal. Of course, there is a problem of accessibility. Sometimes our scientists generate tools but remain inaccessible to users. And we have done our best, like other platforms you have seen in presentation today, to aggregate all this data and decision support tools into one platform, which is called the Freshwater Biodiversity Portal for Uganda, which is a publicly open resource that anyone can go to and access this data, as well as the information for any use, from advocacy, research, capacity building, and others. When we got these occurrences, which we had no access to before, we got excited, and when we saw that it was improving what we know about the fishes of Uganda, we got excited and asked ourselves what it can do at the continental level, and we tried to develop an index for prioritizing areas for conservation for the whole continent. So the index is the aggregating reality of fishes, surface area of water bodies, and conservation status. And what we learn from this index is that yes, these occurrences of species can help us prioritize areas. And we learned from all the analysis that we did that the surface area is the most important factor for prioritizing areas. And that was because the number of species of conservation importance per unit area was higher for smaller water bodies. But that one throws a challenge because some water bodies which are large and are known as very important for conservation are excluded by the index, meaning that we need more data in the water bodies which are large and possibly divide them into smaller segments when calculating the index. We have documented all these efforts comprehensively. So here are different papers that you can look out for reference. They comprehensively document this work that we are doing. So what are the key lessons that we pick? Of course, we learn a lot through this work, but the most important lessons are the ones I'm going to highlight. One is that when you aggregate this data and try to derive decision support tools, you get more understanding of the status of freshwater biodiversity at any scale and the plight that it is facing. And then it is also easier for us to identify or establish critical data gaps that should be explored. And these can be taxonomic, temporal, and spatial. Of course, we are privileged to see increased use of this data in research and other applications. And that is wonderful because that is why it is collected. When we talk about understanding the status and plight of conservation, rather of biodiversity, Here in this pie chart, I have a case for one of the water basins that we have in Uganda. You can see that with this information, you have a better understanding of the conservation status. And here what we see is that we have a large number of species, 62 of them which are threatened. And if you count the data deficient species also as threatened, you see that the challenge is big. Yeah, challenges and opportunities, again, referring to the presentation about data that we had in the plenary, data gaps remain a big challenge. That is, I think, everywhere and at every scale. So with all this understanding, we have been challenged to make sure that we do more work to fill data gaps. But also another challenge that we are facing is that local use of these products can be limited. And that one calls us to do more engagements, do capacity building for data use, to make sure that these products are used for the intended reasons. With that, I would like to thank you for listening.
Thank you again. And the next speaker is Thomas Tomicek, which is myself. And I'm actually very happy to be here today and talk about my project, which was my master thesis and which is also now under review, which is integrating freshwater biodiversity for area prioritization in Colombia. And Colombia is home of one of the most biodiverse freshwater biodiversity in the world, with over 1,600 freshwater species. And it doesn't not only have a lot of species, but it also has a lot of endemic species, with over 374 endemic species. Just here, a tiny glimpse of the vast biodiversity, quite iconic species, for example, the arapaima or the electric eel. And on the other side, you can see the study area, which was not only Colombia here. So I had two study areas, basically. One where I did a spatial prioritization, which is smaller, including also the surroundings countries of Colombia, mainly because rivers do not stop at borders, and connectivity is important to include. And the larger area contained my model domain of the species distribution modeling. And you can see the main macro basins here with the colors, the green, the Amazon, the blue, the Orinoco, the red, the Magdalena Cauca Basin, and two coastal basins which are joined together in the north, the Caribbean, and on the Pacific Ocean, the Pacific Choco. And as we heard already, and I don't want to go into too much detail, freshwater biodiversity also in Colombia is highly threatened, and we see freshwater populations decline is ongoing, also by dams, for example, overexploitation of resources, and many others, and climate change will very likely exacerbate that in the future. And one key aspect to counteract that is usually protected areas. Also in Colombia, there's a vast network of protected areas. Almost 26% of the country are covered by one type of protected areas. However, these were mainly focused on terrestrial species and also quite often set up in remote areas. So you can see, for example, large areas in the Amazon region, and smaller ones in the Magdalena Cauca region, for example. And as also other presenters have already pointed out, there is quite a lack of spatial distributions knowledge about many fish species. So we have the IUCN range maps, but for many species they are data deficient. And this is quite a problem because when we have protected areas, we often don't know if they are even covering freshwater biodiversity. And this hampers our knowledge of these protected areas are actually using our limited resources in nature conservation effectively. And this was my goal to answer for my master thesis. And I used species distribution modeling to then be input for a spatial prioritization. So it's not an entire systematic conservation planning, but part of it. And I used publicly available resources, data resources, which I then also filtered. And my main spatial unit was the subcatchment. So all the species occurrence points were aggregated to the subcatchment to also counteract sampling biases. And I also filtered them by coordinates, names, etc. And in the end, I had almost over 200,000 species point locations, 237 endemic species, 69 were threatened by a type of IUCN threat category, and 29 were both endemic and threatened. And this was then my input for species distribution modeling. This was also already pointed out what it is, so I don't go into too much detail. You have the species point locations, and I used an ensemble model of species distributions modeling to correlate then the environmental factors at the species point locations continuously across space. And here you can see for Pseudoplastoma magdaleniatum where the point locations were, and then the habitat suitability maps for this species. And I did that for 1,313 species. And stacking them on top of each other kind of give you not species richness, but something similar. So the stacked habitat suitability maps, and they actually follow quite nicely the main big rivers in the area. So the Amazon in the south, and the Orinoco river going up here, and its main tributaries. And the lowest habitat suitability we found on the Caribbean coast, which is also where not so many species occur. And using this now to also add some socioeconomic variables, I used for the spatial prioritization the human footprint index, and I used a minimum cost optimization. So the question was, for each species, conserve, for example, 30% of its suitable habitat, which I got from the species distribution modeling, and find the subcatchments with the lowest human footprint index. And also I added on top some constraints. So for example, I wanted to have clumped solutions and not scattered subcatchments here and there, as well as the model prioritized smaller subcatchments compared to bigger ones if they had the same biological information. And I got then first the free choice priority area network, where the model could freely choose the subcatchments with the lowest costs, containing 30% of each species freshwater habitat suitability. And the network was similarly large as the protected area network we see in Colombia. So over 14.4% of the entire study area, which is also larger than containing the surrounding countries as well. And you can see here already, just spatially, that in the Amazon, there were not so many subcatchments selected compared to the Orinoco and Magdalena-Cauca Basin. And compared that now with the protected area network, we can see that the coverage, the overlap between the two is actually quite small with only 25%. And this is quite striking since, again, the two are spatially almost similar. So freshwater biodiversity is not well covered, at least from this modeling perspective, within the protected area network. And from this, the question arose kind of with this sensitivity analysis, were they not selected the protected areas because they were too costly, because they might have high human footprint index, or because they were not containing freshwater fish biodiversity. And actually, we still, when locking in all protected area network in Colombia, we would still need an additional 50% of area outside of the protected area network. So it is not containing this information within them. And comparing that to some IUCN range maps, you can see actually that the model, the species distribution modeling worked actually quite well for some endangered species. And if you want to try out something similar, then I highly invite you to our workshop on Thursday, where you can actually do that. All by yourself. No, we will help you. We will have our expert team again here, which already led this session so nicely. And yes, I hope I could give you then also more details if you want, and you're highly invited. Thank you very much.
Any question? Thank you. I have a question about the prioritization of catchments. So when you talk about prioritizing a catchment, are you talking about the entire catchment? Are you talking about prioritizing like riparian areas across like the rivers? Or because in some of these cases it would be like very unrealistic to actually protect the entire subcatchment.
So my planning unit was actually the entire catchment, which I delineated using GrassGIS tools. So I first delineated a river network using 90-kilometer grid cells, and came up with a stream network and the subcatchments of the entire study area. I had around 38,000 subcatchments for my study area, but they were selected as single units.
Can I ask another quick
Sure.
question? Yeah. So in terms of environmental flows, like in the prioritization, so do you account for what could be happening upstream of the catchments? Because the human footprint is like pretty static in terms of how they measure like the actual variables.
So I included connectivity in my study. So for example, the subcatchments, we calculated the distance between the outlet of one subcatchment and the outlet of another. And this distance information was then included in the prioritization as a cost layer as well.
Thank you, Thomas. Congratulations for your work. I was wondering, because maybe you know that the past year we published the Catalog of the Fishes of Colombia. That is a tool available from the National University of Colombia. And I don't know if, you know, this kind of maps could be very interesting, you know, to link on a taxon page for each species. Are you thinking to make this work like available, like to be can connect this information for each species in this kind of tools?
Yes, so I have all the information per species, so the suitable habitat maps. And yeah, I think we can talk maybe later about it, but sure, that would be possible.
Hi, thank you for the work. I think modeling fish and freshwater species is very challenging. I have two questions. Yesterday we were talking about invasive alien species, and fish is actually one of the biggest threats also here in Colombia. I'm wondering if you were also modeling some invasive alien species that are like fish, invasive alien species that are present here in Colombia, because connected to the previous comment, actually I think that's of high interest for some policymakers here. And the second question is, there is also a big platform for modeling species distributions in Colombia already in place. I don't know what is the current status of them for modeling freshwater species, but I just wonder if you have been in contact with them. Thank you.
Yeah, maybe starting with the second question, I've not been in contact with them, but I know that actually freshwater fish distributions were lacking in what I've seen so far. And yeah, so this would be another great connecting point. And yeah, invasive species, so I checked all my species list with the Colombian freshwater species list, which so yeah, thank you. And we will have our last presentation from Noguera-Urbano, and it will be also about about Colombia. So very excited.
Okay, thank you everybody. I'm gonna switch to Spanish, but in case you need the translation service, you can scan it at the entrance. So yeah, as you wish. So, okay. Hola a todos. Entonces, en este caso les voy a hablar de un proceso de priorización espacial que se llevó en la Orinoquía colombiana, específicamente para ecosistemas de agua dulce, ecosistemas de aguas interiores, en el marco de las metas 3030. Esto fue un proceso participativo desde el principio, con múltiples instituciones, pero destacamos el papel de 3, sobre todo The Nature Conservancy, que nos apoyaron pues, estrechamente en estos aspectos técnicos, porque ellos son autoridad en la Orinoquía colombiana y trabajan bastante en este tipo de ecosistemas. Y después sigue, por ejemplo, la Mesa Nacional de Prioridades, que nos permite como aterrizar nuestros resultados ya a una escala un poco más nacional y articularlo con lo institucional. Y por último, el CIRAP Orinoquía, que pues encierra el conjunto de áreas protegidas de la Orinoquía y que nos permite aterrizar los resultados pues, a una escala un poco más local. Entonces, un poco de contexto de lo que es nuestro marco de trabajo. Primero, pues, estamos trabajando con la priorización sistemática para la conservación. Esto es un marco de trabajo que tiene varios pasos ordenados y estandarizados para seleccionar pues, un conjunto de áreas protegidas, de áreas de interés para la conservación. Entonces, esto permite crear portafolios, pues como ya lo hablaban en la charla anterior, que pues simplemente son mapas que reúnen el conjunto de áreas seleccionadas a través de una unidad de planeación, que es la unidad mínima a ser seleccionada. Básicamente queremos responder a estas 4 preguntas, por ejemplo, en el qué y el dónde están los mapas de especies, que nos dicen pues, dónde hay más especies o ecosistemas y dónde es más importante conservar. Y aquí es importante que no se trata de coger 1000 mapas, por ejemplo, sino escoger los sustitutos de la biodiversidad que mejor me representen esa complejidad ecológica. En el cuánto cuesta esto, pues están los costos. Pueden ser costos monetarios, pero también pueden ser costos ecológicos, como nosotros lo empleamos en este proyecto. Aquí, perdón. Y en cuanto a los targets, pues están las metas globales de conservación, por ejemplo. Esto se usa a través de un algoritmo de priorización que lo que hace es maximizar los costos, maximizar los beneficios y reducir los costos de los portafolios. Entonces nos enmarcamos en estas dos metas, una global del 30% y otra nacional, en donde se busca específicamente para 2030 proteger los ecosistemas de aguas interiores. De acuerdo a esto y pues la importancia ecológica de estos ecosistemas, tenemos dos objetivos. Un primero, pues precisamente desarrollar estos portafolios para estos ecosistemas, pero sobre todo integrando la parte socioecológica y también lo ecológico, ¿no? Porque muchas veces lo social se pasa por alto. Y a partir de esto proponer escenarios que nos permitan pues, articular esto un poco a un nivel un poco más institucional. Bueno, nuestra área de estudio, no tendría tiempo para decirle la complejidad de esta zona, pero básicamente ocupa el 30% de Colombia, lo vemos acá. Si hacemos una radiografía de los ecosistemas de aguas interiores, son estos que vemos acá. Y cabe decir que en los portafolios se suele usar para terrestres como un píxel, pero en este caso usamos una versión de microcuenca, que es cada una de estas unidades que vemos acá. Entonces, en nuestra metodología tenemos insumos que los procesamos a partir del lenguaje de programación de R, sobre todo una librería que se llama Prioritizer, varias cajitas que van a distintos componentes de un problema de priorización. En las características, lo que son los mapas de especies y ecosistemas, más o menos unos 400 mapas. En lo cultural, que esto fue algo innovador en este caso, pues agregamos esos sitios de importancia ritual, sagrados, madreviejas, raudales, pues que están directamente asociados a estos ecosistemas. Hay una charla para el tema de costos por integridad ecológica, los invito a que consulten este ID. Y básicamente lo que nos está diciendo esta capa es que las zonas más costosas son las que están más en rojo. Una aproximación de costos por integridad, pero otra por conectividad, en donde vemos que las zonas menos costosas son estas que tienen un verde más oscuro, que son las que tienen mayor conectividad hídrica en la región. Y cada uno de estos va a una cajita específica que combinando todos estos temas nos dan 144 portafolios que se pueden dividir en aquellos por integridad, de costos por integridad, y otros por conectividad, ¿sí? Y finalmente hay un tema de escenarios, en donde hay un escenario que está restringido a las áreas protegidas, donde se fuerza el algoritmo a seleccionar las zonas que están por dentro de estas zonas, en primer lugar. Y hay otro escenario que es el no restringido, libre elección, en donde el algoritmo puede seleccionar cualquier parte del área de estudio. Entonces, pues como resultados, podemos organizar tantos portafolios en distintas métricas de desempeño con base a estos elementos. Por ejemplo, acá vemos el tema de la eficiencia. En este caso, un portafolio no es eficiente si el target que yo me propongo me está conservando, el portafolio con este target me está conservando más área de la que me estoy proponiendo. Entonces, básicamente este gráfico dice lo mismo, dice esto. Y aquí están unos que seleccionamos que más se acercan a estas metas. Entonces aquí tenemos los que se acercan a la meta nacional y vemos que para todos el porcentaje de cobertura natural es muy alto. y vemos que hay una minoría que es un porcentaje de transformado. Entonces aquí podemos decir que en estas zonas podemos conservar de acuerdo a protección y en estas de acuerdo a, por ejemplo, gestión sostenible. Aquí simplemente un zoom para que vean cómo se traslapan con las zonas, con los ríos principales. Entonces, si vemos, cada uno de los ecosistemas cumplen con la meta del 34%, que es la nacional. Eso nos quiere decir que estos portafolios son suficientes y también íntegros. Y entonces la gran conclusión es que no hay un portafolio perfecto, todo es un costo-beneficio dependiendo de qué quiera lograr. Vemos que la mayoría tienen una integralidad y una suficiencia, es decir, que todos cumplen con las metas y todos están acercándose a esa complejidad, digamos, ecológica del sitio. Pero el tema ya va entre eficiencia y complementariedad, ¿sí? Entonces, ¿qué es la complementariedad? Es simplemente cómo se articula esto con las áreas protegidas o con las figuras de conservación que ya existen. Si ven en estos portafolios, aquí me equivoqué de icono, aquí es el candado cerrado, pero si ven estos portafolios, estos son los portafolios donde se fuerza a las áreas protegidas a conservar dentro de estas zonas. Entonces aquí, por supuesto, la complementariedad es del 100%, pero aquí ese sobrelape empieza a ser un poco menor. Sin embargo, encontramos que incluso cuando elegimos libremente, pues esa complementariedad no es tan baja y es del 44%. Sí, entonces, pues, es ese trade-off entre lo que es eficiencia y complementariedad. ¿Qué sigue? Entonces, pues la idea es que esto es un proceso piloto para todo el país. Se comenzó la Orinoquía porque es una región bastante compleja en estos ecosistemas, pero la idea es completarlo para todas las regiones de una forma un poco más estandarizada y pues automatizada. Estamos creando un repositorio en GitHub y por supuesto pues compilando todos estos saberes en un artículo científico. Cualquier pregunta, pues aquí tienen mi contacto y pues yo también los puedo comunicar con todo el equipo de trabajo. That's it for today.
Thank you for a really comprehensive presentation and work. So I have possibly a few questions that it's more about clarification. So you have done more than 100 scenarios, right? And you try also to get, I want to know if the scenario result has been validated. So when you run the scenario, get the result, and you make your final decision for prioritization, did you manage to validate where the model and the scenario they are confirming? That's one. Another thing is related to scenario. If you can possibly highlight one of the potential scenario which show more impact, positive impact on species, how did you manage to link scenario activities and species impact?
Okay, I'm going to try to answer this in English, so just be patient.
Please.
So, yeah, we have a lot of portfolios. So we evaluate and assess a lot of performance metrics, but more important is the expert knowledge, you know. So we meet with a lot of local experts to validate our results. So in this case, can we switch to the slide? In this case, we prefer the connectivity-based scenarios because there are more approach to what we need and because they reflect more the hydrological complexity of the study area. So yeah, I think that's the answer, but yeah, just to clarify this portfolio. And we prefer not to lock in to protected areas. We prefer that the algorithm chose freely. So yeah, that's better for us, and it's better for the dynamic of the study area. I think that's it.
Maybe to wrap up the session, I want to thank again all the presenters and also the audience. I think we've seen now in the second, this afternoon, a nice example